Hello, and welcome back to the Prentice Hall Biology textbook. Today we'll be covering Chapter 13, Genetic Engineering. Okay, 13-1, Changing the Living World. So, to change the living world, humans use a number of methods, including selective breeding. So selective breeding is allowing only those animals with desired characteristics to produce the next generation. So humans use selective breeding, which takes advantage of naturally occurred genetic variation in plants, animals, and other organisms to pass desired traits on to the next generation of organisms. Nearly all domestic animals and most crops have been produced by genetic engineering. So under selective breeding, there's hybridization, which is the crossing of dissimilar individuals to bring together the best of both organisms. And then we have inbreeding. Inbreeding is the continued breeding of individuals with similar characteristics, and it's used to maintain the desired characteristics of a line of organisms. However, it does have its risks. If there are two recessive alleles, that can cause genetic defects. And then we have increased variation. So breeders can increase the genetic variation in a population by inducing mutations, which are the ultimate source of genetic variability. So, but we produce most of the mutations in bacteria to produce new kinds of bacteria. Millions of bacteria can be treated with radiation or chemicals at the same time, increasing the chance of a useful mutant. And then also, using variation, we can produce new kinds of plants, known as polyploidy plants, which we've talked about earlier, and those are just larger and stronger plants. Okay, 13-2, manipulating DNA. So until recently, animal and plant breeders could not modify the genetic code of living things. Scientists, however, could use their knowledge of the structure of DNA and its chemical properties to study and change DNA molecules. Different techniques are used to extract DNA from cells, to cut DNA into smaller pieces, and to identify the sequence of bases in a DNA molecule. And this allows us to make unlimited copies of genetically engineered DNA. So the tools of molecular biology. So the changing of molecular codes is very similar to rewriting computer code. It's changing the base pairs, or changing the base sequences in a molecule of DNA. So genetic engineering is the making of changes in the DNA code of a living organism. So the first tool is DNA extraction. So it's a simple chemical procedure where the cells are opened and the DNA is separated from the other cell parts. And then there's cutting DNA. So when DNA molecules are too large to be analyzed, they are cut into smaller fragments using restriction enzymes. And hundreds of specific restriction enzymes are known, and each one cuts DNA at a specific sequence of nucleotides. And then we have separating DNA. So using the process of gel electrophoresis, which is a mixture, a mixture of DNA fragments is placed at one end of a porous gel, and an electric voltage is applied to the gel. When the power is turned on, the DNA molecules move towards the positive end of the gel because they're negatively charged. So the smaller DNA fragments move faster and farther than the larger ones. And gel electrophoresis can be used to compare the genomes or gene composition of different organisms. It can also be used to locate and identify one particular gene out of the thousands of genes in an individual's genome. Next, we have using the DNA sequence. Once DNA is in a manageable form, its sequence can be read, studied, and even changed. So reading the sequence. A single strand of DNA with unknown bases is placed in a test tube with DNA polymerase. That is an enzyme that copies DNA, and a large amount of the 4 nucleotide bases, adenine, thymine, cytosine, and guanine. And some of these bases have been stained with dye. The enzyme creates a complementary strand of DNA, and each time a dyed base is synthesized, the synthesis is terminated. So this creates fragments of different length. And then using gel electrophoresis, and the order of colored bands tells the exact sequence of bases in the DNA. Okay, then there's cutting and pasting. So DNA sequences can be changed in a number of ways. Short sequences can be assembled using laboratory machines known as DNA synthesizers, while synthetic sequences can then be joined to natural ones using enzymes that splice DNA together. And then recombinant DNA is DNA molecules produced by combining DNA from different sources. So making copies. DNA copies are made using a technique known as a polymerase chain reaction. So scientists add primer to portions of DNA, and these primers tell the DNA polymerase where to start working. The DNA is then heated to separate the strands, and the complementary strands are created. When the complementary strands are separated, they keep on more strands are synthesized. And then the more complementary strands are made, the more original copies we have of the DNA. Okay, 13-3, cell transformation. So during transformation, a cell takes in DNA from outside the cell. This external DNA becomes a component of the cell's DNA. So Griffith's experiments with the lethal and non-lethal strains of pneumonia were a form of transformation, and those showed that cells could take in DNA from the outside. Okay, transforming bacteria. So to transform bacteria, foreign DNA is first joined to a small circular DNA molecule known as a plasmid. And now plasmids have two essential features that make them very useful for DNA transfer. It has a DNA sequence that helps promote plasmid replication, and it has a genetic marker, a gene that makes it possible to distinguish bacteria that carry the plasmid and the foreign DNA from those that don't. Okay, transforming plant cells. So using recombinant plasmids to infect the plant cells. The cell walls are removed, and the plant cells in the culture will sometimes take up DNA on their own. If they don't take up the DNA on their own, the DNA can be injected directly into the cells. If the transformation is successful, the recombinant DNA is integrated into one of the chromosomes of the cell. Alright, now transferring animal cells. They can be transformed in some of the same ways as plant cells. Most egg cells are large enough that DNA can be directly injected into them. Recently, it has become possible to eliminate particular genes by careful design of DNA molecules. So using ends that will combine with specific sequences in the host chromosome. The host gene normally found between these two sequences is lost or specifically replaced with a new gene that is inactive. Okay, 13-4, applications of genetic engineering. So genetic engineering makes it possible to transfer DNA sequences, including whole genes, from one organism to another. So for instance, we were able to take the glowing gene from a firefly and inject it into tobacco plants, which resulted in glow-in-the-dark leaves. So transgenic organisms. So transgenic means it contains genes from other species, and genetic engineering has made it possible to construct transgenic organisms. Genetic engineering has spurred the growth of biotechnology, which is a new industry that is changing the way we interact with the living world. For instance, we've now created transgenic microorganisms. So transgenic bacteria now produce a host of important substances useful for health and industry. Diabetes patients are now treated with human insulin produced by human genes inserted into bacteria. We've also created transgenic animals. Mice have been produced with human genes that make their immune system act similar to those of humans. And this means we can test out different types of cancer research or cancer drugs on them. And then some transgenic livestock have extra copies of growth hormone, which means they grow bigger and can quicker. And then we have transgenic plants, which we've talked about before with the polyploidy plants. Also, then cloning is a very controversial topic under genetic engineering. So a clone is a member of a population of genetically identical cells produced from a single cell. So clone colonies of bacteria and other microorganisms are easy to grow. However, in 1997, a Scottish scientist, Ian Wilmot, cloned a sheep named Dolly. And this accomplishment was highly controversial. There were many different scientific debates on the morality of cloning and what it meant for the humans and cloning themselves. Okay, key concepts. So, for the key concepts, be able to give one example of selective breeding. Relate genetic variation and mutations to each other. Describe the process scientists use to manipulate DNA. What is transformation? How can you tell if a transformation has been successful? What is a transgenic organism? List one practical application for each of the following: transgenic bacteria, transgenic animals, and transgenic plants. Alright, that's it for chapter 13.